Hello YouTube, this is Sam from Tigers Schooling. In this section of the video, we're gonna talk about apple. Wait, what? No, we're not gonna talk about apple, but we're gonna talk about something that is related to the apple. Like why you need an apple, like why we need food, right? Ever thought about it? It's totally because we need energy. So how we get energy, for example, if you're eating an apple? Consider it that way. For example, in that section, we'll be starting from an apple and we'll end up at ATP. So we need an apple to get energy, and our body utilizes energy in the form of ATP, and we'll understand the whole process using the electron transport chain. So be with me, because in the end, we are going to understand everything. To be honest, electron transport chain I'd make it that much easier. You will be a genius in your class. So stay with me till the end in order to understand the whole concept. Consider it that way. For example, I have got an apple. So apple is my food, right? So I'll go write it down like this. So this is my food. This one is apple, right? So to convert to food, what we do? The apple goes through the citric acid cycle. It is citric acid cycle, right? Which is also called TCA, or you can say the Krebs cycle. So in the Krebs cycle, like we got a food, which the food is going through the citric cycle, or you can say TCA cycle, and the TCA cycle convert our food into the stored form, like an energy stored form, and that is, it is going to convert this food into the NADH. Probably it is going to convert this food into the NADH, which is going to one citric cycle going to make 3 NADH plus 1 FADH2 and 1 GTP, right? Very simple. So these things are actually stored energy, but we still don't need energy in this form. We need to convert these guys, because body does not utilize the energy in the form of NADH or FADH2 or GTP. We need energy in the form of ATP. So to get an ATP, we put these guys. These guys must go through a process or a chain, which is called as the electron transport chain. So they go through the electron, which is E, T means transport, C means chain, and the electron which these guys have is going to pass through series of complexes, which we're going to talk in just a bit and make it very much easy. And finally the electron will pass through that whole chain, and finally it gives us finally we go through the oxidative phosphorus relationship and we get our ATP, which is our energy molecule. Very much easy. Look, we started from food, which was an apple. We gone through the citric acid cycle. We got these stored energy molecules from one citric acid cycle. And finally, we go through the electron transport chain. And there's a small step, which we'll also discuss in the electron transport chain, but in our next video, which is oxidative phosphorus relationship, which is related to the electron transport chain. Not that much complex. Very much easy. So let's get started and start the lecture on the electron transport chain. In order to understand the electron transport chain, we must have an idea of the mitochondria. This diagram is my diagram. I know I made it very rough. You people might not like it, but I'll try to explain it the way it is. All right. So this is mitochondria. We know mitochondria has two layers. The outer layer. So this one is the outer layer, or you can say outer membrane. This one is the outer membrane, and this guy, I made a double layer, right? And this guy is inner membrane, right? And these are lipid bilayer, which are kind of, these, I don't know what you call them. These green color, what I mentioned, or these hanging structures are actually the lipid bilayer. So the inside one is the inner membrane, mitochondrial membrane, and in between the outer and inner mitochondrial membrane, we got a space, right? This is space, this empty space. I'll just highlight, I'll just use this one, this color, and this space is called intermembranal space. Right, very much easy. We need to have a basic idea of everything. In the final stage, we'll be understanding. We're going to, and then we're going to talk about the process. First, we need to understand the basic. Outer membrane, this one inner membrane, and this one is the, inner membrane, intermembranal space, and the outer membrane. And inside one, this one is the matrix. You know, the matrix inside the mitochondria. So this one is the matrix. So what happens? You eat a food. Food goes into your mitochondria, and over here the TCA cycle occurs in the matrix, and in the matrix you get all those little stored energies that are NADH, which are 3 molecules, you get FADH2, which are 2 molecules, and you finally get the GTP. And these boys, or you can say NADH and FADH2 and GTP, again will go through the electron transport chain and give us our ATP boy, right? So what happens? We got the structure, we understand everything. Now I'll put this structure. What I'm going to do, I'm going to cut this diagram like this. I'm going to cut it from here and cut it from here, and just going to paste it over here on my big picture. So cutting it from here and cutting it from here, and just putting it the way over here. So we done it, we put it over here, and now I have something new. Look, we got the outer membrane. This one is the outer membrane, this one the inner membrane, and this space, which is in white color, or I'll just put the right in green. This space is called intermembranal space, right? And this is the intermembrane. This is the membrane itself. It's the space which is between the outer membrane and the inner membrane. But look at the inner membrane. We got few really special structures. What these guys are doing out here. So these guys, which are embedded, you can say, because they are embedded in the inner membrane, are actually the complexes. Look, the word complex seems so complex that people or students, when heard about the electron transport chain, and when they hear about the complex, everything seems complex. You know what I mean by the complex? Like you kind of don't understand it. But complex are actually nothing. They're simply proteins. Get it? So these guys are protein. I'm not going to use the word complex. When I need it, I use it. But just to make it easy, remember, these guys are proteins. So we are actually naming these guys like this protein, this protein, this protein, this protein. Electron transport chain has 4 important proteins. So we call these proteins complexes. We just call them. That's it. This is complex 1, this is complex 2, this is complex 3, this is complex 4. That's it. We are going to name them. So they are actually proteins, but we want to give them a name. So this guy is complex 1, complex 2, complex 3, complex 4. But these guys have another name. I'll just talk about it in just a bit. All right. We're clear about this boy. We're done with the complexes. So what happened? This one is the complex 1, complex 2, complex 3, complex 4. And this guy, which we'll use in the oxidative phosphorus relationship, we'll talk about that one, because this guy is going to generate our ATP, and in that video we'll be talking about the oxidative phosphorus relationship. So this is the intermembranous space. So remember the matrix, where our NADH and where our food was converted in the matrix. This area is the whole matrix, right? Our food was converted into the NADH, or you can say another, like we got 3 molecules, probably, yeah, 3 molecules of NADH and 2 molecules of FADH2, like, I'm sorry, 1 molecule of FADH2, and we got 1 molecule of GTP. So what happened? This, we'll talk about the process. This NADH guy is going to give an electron. An electron is kind of hanging around over here and down there, and trying to pull the hydrogen from here and putting them into the intermembranous space. I'm going to talk about that. Now let's talk a little bit detail about these complexes, and finally we're going to talk about the process. So we must understand the four complexes, what they have inside. Actually, they have actually coenzymes as well. So four complexes, but we already have two other structures. There is this one, this guy, which is a coenzyme Q, which is kind of hanging around the complex 2, and or you can say between complex 2 and 3. And there's another coenzyme which is between complex 3 and 4, which is cytochrome C. Remember, every complex and every coenzyme, they are protein in nature except this guy. This Q guy is lipid in nature. If somebody is going to ask you what are the complexes and structures inside this electron transport chain made of, you will be saying they are made up of they are protein in nature, except the coenzyme Q, or you can also call it the ubiquinin. They also call it the ubiquinin, this small structure. Now let's talk about something else. Now let's specify, or you can say talk in detail about every complex, or say every protein. So this protein is actually a dehydrogenase. So these 1 and 2 you'll be talking about, these guys are actually dehydrogenase protein, right? So these are dehydrogenase, right? And this guy out here, the 3 and 4 are actually the cytochrome. So we call them cytochrome, right? These are cytochromes. So what happens? This one, the both are dehydrogenase, but this guy is actually the NADH dehydrogenase, right? This guy is called succinate dehydrogenase. Succinate dehydrogenase. Both are dehydrogenase, but this guy is this guy, the complex 1 is also called the NADH dehydrogenase. The complex 2 is called succinate dehydrogenase, and the complex 3, which were cytochromes, we call them, the complex 3 is called cytochrome BC1, and this number 4 guy is called cytochrome AA3. Very simple. If somebody is going to ask you what is the alternative name of these complexes, you would say NADH dehydrogenase, number 2 succinate dehydrogenase, then number 3 is cytochrome BC1, and finally we got the cytochrome, which is complex 4 is the AA3. Very much easy. Now we have to remember a few things more. These complexes also have coenzymes attached to them, right? These complexes have coenzymes attached to them. Remember this one. So this guy have a coenzyme which is iron sulfur, right? Iron sulfur, the iron sulfur or coenzyme, and again this one, iron sulfur, and again this one, they have again the BC1, right? Okay. So these are the common common coenzymes which are attached to these. The NADH dehydrogenase has a common iron sulfur containing, and this guy number 2 also has the iron sulfur containing coenzyme with it, succinate dehydrogenase, and this one as well. But two other structures which are present with this boy, like this guy has another structure which is called FMN. Like these structures are important because these kind of help in transferring our electron or carrying over electron from one place to another. That's why we need these structures. So other structures, succinyl dehydrogenase has the FAD. That's it. Iron sulfate all have common iron sulfate except this boy AA3. We haven't given anything to them, but FMM only specifies to this guy, which is NADH dehydrogenase. Succinyl dehydrogenase only has FAD. But the last guy, which was the cytochrome AA3 complex 4, was thinking, why you haven't given me anything? So we say, just wait a minute, we'll give you something called copper. We will give them the copper. So what happens? We're going to give him the copper, 2 coppers, we're going to give him the copper A and copper B. That's it. You got copper A and copper B. Very easy, very much easy to understand. So this is all about the structure. We're not going to add up any new thing. So if you are thinking, oh my god, what a mess I have made, and I'm going to put all other structures, no, there is nothing else. This is it, very much easy. We got 4 complexes, means 4 proteins, nothing so much. We just name them, we call them complex 1 NADH dehydrogenase, succinyl dehydrogenase. Number 3 we call it cytochrome BC1, and number 4 we call the cytochrome AA3. But these boys have something, like you have to have something which actually helps in transporting your electron. So we give them the three complex have something in common, that is iron sulfate. But the 4 guy has only few things, they haven't got anything, so we give them the copper A, you just take it, copper B, that's it. And for the complex 1 we have given a special thing, that is FMN. Complex 2 we have given him the FAD. So this is the basic structure of the whole inner membrane of the mitochondria, and all the complexes present here. Now we'll be going to talk about one thing more. These are the coenzyme which are attached extra. These are cytochrome C and Q, which are actually also going to help in the transfer of electron. So now let's talk about the process. So before we're gonna talk about the process, we start from here, the word electron transport chain. So electron transport chain means we are actually transporting the electron. Where does the electron come from? We're gonna get the electron from this guy. They have stored energy, like electron in their shells has the highest energy. So the NADH guy is actually come here, over here, NADH, and this guy has an electron in the hydrate form, H negative. So that's why what happens in this region, in the complex 1, the NADH dehydrogenase actually going to convert. So we have got NADH, I write it down again. So this NADH dehydrogenase is actually going to convert into the NAD, right? And taking this boy's electron, what he's going to do, he's going to take this boy's electron and pass it, and take this boy's electron and pass it from the complex 1 to again, and as it crosses the complex 1, it moves towards it goes through small things, like from the, as we take electron, it goes from NADH dehydrogenase to FMN, right? I'm sorry, I just gone through that one. It goes to FMN, and from FMN it goes to iron sulfate, and from here it just transfers, like it has to pass through this series and moves towards the cytochrome Q, which is the coenzyme Q, which is also called as the ubiquinone. So we take an electron from the NADs through the NADH dehydrogenase, which passes to FMN and iron sulfate, and which finally goes to the ubiquinone Q. And by transferring this electron, why we are actually transferring this electron? So the energy which it has lost actually helped us to move the proton. Like how many protons are actually going to move, right? So I'll just use another color marker. This one, I guess, yeah. So what happens as electron crosses the complex 1? The energy which it loses, or the energy which it has given, will be utilized to take the hydrogen which are present in the matrix. Like there are abundant hydrogen present in the matrix, which actually come from here. We'll talk about that one later. So we take these hydrogen outside into the intermembranous space. So as the electron crosses complex 1, how many hydrogen ions, or you can say hydrogen protons, are going to pump through the complex 1? You would say 4 hydrogen, right? 4 hydrogen are going to actually cross this one, and finally the electron reaches to the coenzyme Q, where it is stored. Now it's the turn of the complex 2. Complex 2, what it does, like complex 2, what actually it does, it actually takes this complex Q, actually convert, and another which convert the succinate, I'm not writing it in detail, succinate into the fumarate, which actually gives an electron, right? Which gives an electron, I'll just put it like this one, which gives an electron, which first goes through the succinyl dehydrogenase, like we use an enzyme which is succinyl dehydrogenase, and this guy is going to transfer electron to FAD, and FAD is going to transfer it to the sulfur iron sulfate, and finally this iron sulfate is going to transfer the electron towards the ubiquinone Q, or you can say the coenzyme Q. So over here we get an electron, from here we get an electron, but remember this one, this guy has much more energy and this process is more efficient, so this gives us 4 hydrogen. Like this boy from complex 1 actually crosses the 4, we get 4 hydrogen pumped over. But in this process, like when we go to the complex 2, we get none hydrogen, like no hydrogen is going to be pumped because there is not much enough energy. Right, we're clear about that, like we don't have enough energy, that's why we are not getting any hydrogen in the complex 2. That's why it's not that much useful. And from here, what happens from ubiquinone Q, or you can say cytochrome Q, or like coenzyme Q, the electron actually transfers from here. We're going to take the electron and let it move towards this guy, which is complex 3, or we call it the cytochrome BC1. So from the cytochrome BC1, the electron actually going to transfer, it comes towards this boy, which is the iron sulfate over here, and electron from iron sulfate, it moves toward the cytochrome C, which is over there, and the coenzyme, like this guy is kind of playing around, like, right? So this guy is going to cytochrome C, and from cytochrome C, it is again going to enter into the complex 4. That is the electron again is moving. So that is the complex 4, which is moving downward. So this complex, which is the cytochrome AA3 complex 4, we call it. Over here, the electron come towards the copper ions, or the copper A and copper B. From here, the electron is actually released, or actually used, or actually you can say the electron from here move and attaches itself with hydrogen and oxygen, right? Oxygen and hydrogen are kind of wandering inside the matrix, so they kind of join together with them and convert them into water. Like we need 1 water molecule and plus 2 hydrogen ions and 2 electrons to make 1 water molecule. So 1 water molecule H2O, like we need 2 electrons, 2 hydrogen and 1 oxygen molecule to convert this boy, the ending electron, the 2 electron, into the water molecule. Right, very much easy complex, very much easy idea to understand. One thing more I just forgot to talk about is, as the electron crosses from this one towards cytochrome C, it actually also pumps 4 hydrogen. Like in complex 1, we pumped up 4 hydrogen. From here again 4 hydrogen ions are going to be pumped over into the intermembranous space from matrix. Remember this one, we are taking hydrogen from matrix and moving towards the intermembranous space. There's a reason behind it. And from cytochrome C, as it enters this guy, the complex 4, as it crosses the complex 4, again it pumps two other hydrogen molecules. So in total there are about 10 hydrogen ions, protons you can call them, are actually driven from inside the matrix toward the intermembranous space by taking 1 electron, or by taking the electron by our 1 electron transport chain. So the our goal is actually to make a hydrogen or proton gradient. Like we are continuously taking out the hydrogen, like we're continuously taking out the proton and putting them into the intermembranous space. Why we are doing that? Because as we reach a threshold that inside the matrix there will be less protons, and in the intermembranous space there will be more protons. So what happens? So these protons, as we collect them over here in the intermembranous space, these protons actually we're going to use these protons, these 2 3, which are 10 in number now, but they increases repeatedly, the electron transport chain goes through these. There's a gradient which is formed. So these have more potential. What happens? They go through another complex, which is we'll talk about in next lecture, which is the oxidative phosphorylation. And these guys go through this ATP synthase, or the complex 5 sometimes they call them, but it's not included in the electron transport chain. If somebody is going to ask you about electron transport chain, Till here our electron transport chain ends, but I'm telling you, just giving you an idea why what actually we are going to do. So these guys collect over here. So these hydrogen, then afterward there's a gradient, like there's a more potential. These hydrogen going to pass from the more gradient, or where the more amount of the proton, they're going to move against the gradient, not against the gradient, towards the gradient, toward the lower where the less hydrogen is, less proton are present. They go downward. So as 3 molecules go downward, we get 1 ATP molecule, like 1 ATP molecule is formed. So the whole energy actually came from the electrons. Electron gave it to the help in forming the gradient, like it gave to the hydrogen, and hydrogen forming again, it gives us the finally 3 protons pass through this molecule, you can ATP synthase, and finally giving us the ATP. I will be talking about the oxidative phosphorylation, or the formation of ATP, in our next video. Please make sure to visit our site, that is www.tygschooling.com, to read our latest blog and help us to make our channel, our website better, and to make it more reachable. Please keep visiting Tyg's Schooling. Thank you.